Good evening, students. Today, under chapter Human Health and Diseases, we are going to revise the topic cancer. So, let's begin. Cancer. Now, the normal cells which are there, their growth and their differentiation into different tissues are controlled and regulated by our body. So normal cells they show the property of contact inhibition. ये जो cells होते हैं वो जब surrounding cells के contact में आते हैं so they stop dividing. This is contact inhibition. So you can learn the definition here. Now cancerous cell they lose this property and due to that they keep on dividing at an uncontrolled rate and give rise to masses of cells. These mass of cells are known as tumors. Tumors are divided into two types, benign and malignant tumor. In exam, you may get question like differentiate between benign and malignant tumor. So, the benign tumors they are localized tumor, they do not spread, whereas malignant tumor, they proliferate from one part of the body to rest. Benign tumors, these are the masses of cells which are covered with connective tissue layer which prevents them from spreading. Whereas malignant tumor, they do not have such connective tissue sheath around them. So once the mass of cell, the tumor, it increases in size, the cells they detach from that tumor and through the bloodstream they get lodged to other part of the body causing secondary tumor at that place. This property of cancerous cell is known as metastasis. ठीक है तो मेटास्टेसिस क्या है the cells they detach and they reach to other parts of the body through blood forming and thus forming new tumors over there. What are the causes of cancer? So cancer causing agents they are known as carcinogen. There are different agents like physical agent. Physical agent में आते हैं like the patients who are having long history of X-rays, they might have the chances of getting the cancer. Okay, because X-rays they cause mutation in the DNA of the patient. So ionizing radiation like X-rays, gamma rays, non-ionizing radiation like UV rays, they causes DNA damage and thus leading to neoplastic transformation. Then there are chemical agents. These are the chemical carcinogens like the chemicals present in tobacco, which causes lung cancer, mouth cancer, etc. Then there are biological agents, living organism which causes cancer. So example of that is human papilloma virus which causes cancer of uterus. Now, normal cells, all normal cells, they have cellular oncogenes which are also known as proto-oncogenes. Now, these oncogenes, due to any of these agents, when they get activated, they causes the oncogenic transformation of the cells and the cells, they loses their property of contact inhibition and results in the development of cancer or tumor. How cancer is detected? The cancer is in the tissue. So it can be detected with the help of technique that is biopsy and histopathological studies. बायोप्सी में हम क्या करते हैं जो ट्यूमर है उससे थोड़ा सा टिश्यू हम एक्सट्रैक्ट करेंगे देन इट इज कट इनटू स्मॉल सेक्शंस स्टेन्ड एंड ऑब्जर्व्ड अंडर द माइक्रोस्कोप so histopathological studies are done by the pathologist. So that is for tissues. Now, if the cancer is of blood, that is leukemia, so in that case, we are performing blood and bone marrow test to detect such cancer. Cancer of internal organ is detected with the help of X-ray, CT scan और MRI scan. CT scan में हम क्या है X-rays को यूज करके 3-dimensional image प्रोड्यूस करते हैं internal organs की और MRI scan में strong magnetic fields and non-ionizing radiations are used to detect the pathological and physiological changes which has taken place in the tissue of an organ. Now, there are some cancers. In the detection of that, we make use of antibodies. So, antibodies against cancer-specific antigens are also used for detection of certain cancers. Some cancers, they are genetic, they are transmitted from parents to their children. So, in that case, we make use of molecular biology technique to detect them. Coming to treatment of cancer, how it is treated. So there are some common approaches like surgery, radiation therapy, and immunotherapy. In radiation, जो ट्यूमर सेल्स होते हैं उनको इरेडिएट करते हैं विद द रेडिएशन, ठीक है, so that kills the cancerous cells and doesn't harm the normal cell. Same way in chemotherapeutic drugs, the chemicals are injected in the body and that kills the cancerous cells. In most of the cancers, combination of these therapies are used like surgery. पहले surgery से tumor remove करते हैं, फिर radiation देते हैं. Chemotherapy is also given in order to kill the cancer cells. For treatment, we also make use of biological response modifiers like alpha interferon. तो ये क्या है? जो ट्यूमर सेल्स होते हैं कई बार वो डिटेक्ट नहीं हो पाते और हमारा इम्यून सिस्टम उसे डिस्ट्रॉय भी नहीं कर पाता है, तो हम पेशेंट को ये अल्फा इंटरफेरॉन बायोलॉजिकल रिस्पांस मॉडिफायर जो है वो इंजेक्ट करते हैं. ये अल्फा इंटरफेरॉन उनके इम्यून सिस्टम को एक्टिवेट करेगा एंड हेल्प्स इन द डिस्ट्रक्शन ऑफ द ट्यूमर. अब बोर्ड क्वेश्चंस देख लेते हैं रिलेटेड टू दिस टॉपिक. How is cancerous cell different from normal cell? Cancerous cell कैसे normal cell से differ करते हैं? तो normal cells जो हैं, they divide at a controlled rate. They have the property of contact inhibition. Cancerous cells, they loses that property. ठीक है? So there is no adherence in cancerous cell, whereas normal cells remain adhered to one another. Tumor is formed in cancerous cells due to repeated uncontrolled cell, whereas normal cell, their division is regulated and controlled. ठीक है, cancerous cells की जो है कोई definite lifespan नहीं होती है, whereas normal cells have definite lifespan and old cells are replaced by new cells. Second, explain what is meant by metastasis. तो हमने पढ़ा था metastasis क्या है? Cancerous cells जो ट्यूमर है उससे जो सेल्स होते हैं, दिस लॉस ऑफ दे थ्रू द ब्लड स्ट्रीम दे गेट लॉज टू the other tissues and organs present in the body and causing secondary tumor over there. So this property of cancerous cell is known as metastasis. Differentiate between benign and malignant tumor. This we already discussed. ठीक है? All human beings have cellular oncogenes, but only a few suffer from cancer disease. Give reason. तो cellular oncogene हम सभी में होती है, लेकिन वो inactive होती है. Due to the different carcinogenic agents, these genes they might get activated and thus resulting in oncogenic transformation. How is malignant tumor differ? So already हमने डिस्कस कर लिया कैसे malignant tumor benign tumor से differ करता है. Fifth question, why alpha interferon is given to the cancer patient? Alpha interferon क्यों दिया जाता है? ठीक है, alpha interferon क्या होते हैं? Biological response modifiers and इनमें ability होती है कि ये हमारी immune system को activate करे, तो हमारा immune system cancerous cell को destroy करेगा. So that's why we biological response modifier, that is alpha interferon, is given to the cancer patient. Question number 6. Cancer is one of the most dreaded disease of humans. Explain contact inhibition and metastasis with respect to the disease. तो दोनों की आपको definition लिखनी है. Contact inhibition is the property of normal cells. These normal cells, when they are in contact with other cell, their growth is controlled. They do not divide at an uncontrolled rate. ठीक है, metastasis property होती है cancer cells की कि they can detach from the tumor and reach the distant sites in the body and cause cancer over there. Name the group of genes which have been identified in normal cells that could lead to cancer and how they do so. तो ये सेम रिपीट हुआ है. Group of genes, proto-oncogene और cellular oncogenes. And ठीक है, फिर how they causes cancer. So when they get activated due to some certain conditions, due to certain carcinogens, so they causes oncogenic transformation of the cells. Name any two techniques which are useful to detect cancer of internal organs. So, internal organs में क्या था? CT scan, MRI scan. ठीक है? Why are cancer patients often given alpha interferon as part of the treatment? Already discussed. Question 7. What happens to a normal cell in a body when oncogenes get activated under certain conditions? So, क्या होता है? They loses their property of contact inhibition. Oncogenic transformation occurs in these cells and they start multiplying at an uncontrolled rate, forming the tumor. Which technique are useful to detect cancer of internal organ? Again repeat हो रहा है. CT scan, MRI scan. Yeah. Then why are cancer patients often given a alpha interferon during their treatment? So you can see ये alpha interferon जो biological response modifier है, this question is asked again and again in the exam. So prepare it thoroughly. So that is all about cancer. Hope you must have understood the topic. So see you in the next class. All the best for your example. Thank you everyone.